Alright, welcome back. So, the verse of the day is, my dear brothers and sisters, take note of this. Everyone should be quick to listen, slow to speak, and slow to become angry. James 1:19. Alright, with that we shall start. So this is the structure of a virus. Capsid is on the outer layer, nucleic acid is on the inside, and this is so this one is a naked virus, and this is an enveloped virus, and enveloped viruses have a surface protein, which are these little projections. Okay, that means it's enveloped, and enveloped virus with a helical capsid. This has this means that the virus is enveloped, and this one has a helix in the center, and that helix has viral RNA. Viral RNA. And bacteriophage is something that eats bacteria. Okay, this is the structure of it. Viral genetics. So we're going to talk about this viral genetics. This is how viruses replicate. So, recombination, exchange of genes between two chromosomes by crossing over within regions of significant base sequence homology. So you have figure A and then figure B, which makes figure C. It's a mix of A and B. See, blue and green. Okay, all right. So what this means is the mixing up of DNA and proteins to make a new progeny of viruses. So a progeny is like a child. It's a love child of viruses. So that's what recombination is. It's a mixing of DNA and proteins of two viruses to make a new virus child. Exchange of genes between two chromosomes by crossing over within regions of significant base sequence homology, mixing of DNA and proteins to make up a new progeny, children of viruses. Okay, reassortment. What's reassortment? The viruses with segmented genomes, like the influenza virus, exchange genetic material. For example, a 2000 novel H1N1 influenza a pandemic emerged by a complex viral reassortment of genes from human, swine, and avian viruses has potential to cause an antigenic shift. Okay, so you see this right here. When viruses with segmented genomes, what are genomes? Segmented genomes are segmented RNA or DNA. Okay, like the influenza virus, exchange in genetic material, like you see here. Okay, this is a novel like the 2000 H1N1 influenza virus. They switch, they give parts of their DNA to their child, which is right here. See this blue and green? This one's all blue, this one's green, this is blue and green. This is a reassortment. So, an influenza virus, it's made up of, so this right here, it says a pandemic emerged by a complex viral reassortment of genes from human, swine, and avian viruses. What that means is influenza virus is made out of humans, pig, and birds, and that's what caused a pandemic. Okay, it has potential to cause antigenic shift. Yep. Okay. Okay. So that's what that is. Complementation. When one of two viruses that infect the cell has mutation, it results in a non-functional protein. The non-mutated virus complements mutated one by making a functional protein that serves both viruses. For example, hepatitis D virus requires the presence of a replicating hepatitis B, a virus to supply HBsAg, the envelope protein for HDV. Alright, so when one of the two viruses that infect a cell has a mutation that results in a non-functional protein. Okay, so one of the two viruses that infect a cell has a mutation results in a non-functional protein. So this one is non-functional. This blue and figure red want to come into your body and kill you. Okay, so one is functional. The other one is like it has the tools to kill you. The other one does not. The red does not have the tools to kill you. Alright, when one or two viruses that infect a cell has a mutation that results in a non-functional protein, the non-mutated virus, non-mutated virus, figure blue, complements the mutated one by making a functional protein that serves both viruses. Interesting. Okay, for example, hepatitis D virus requires the presence of replicating hepatitis B virus to supply HBs antigen, the envelope protein for hepatitis D. Okay, so this blue one is working fine. This blue virus is working fine. The red virus does not work. So what does the blue virus do? It says, "Don't worry, homie, I got you." And then it makes a blue cover and protects the red one. So now it becomes functional. Just like that, hepatitis D, for hepatitis D to survive in our body, hepatitis D needs hepatitis B. Okay. Yeah, that's what that means. So in our body, the functional one would be hepatitis B. This would be hepatitis D. And this functional one would, like now hepatitis D would be functional because it has hepatitis B protecting it. Okay. So so what they're saying is you need hepatitis B virus to be infected with hepatitis D virus. Hepatitis D virus is non-functional when hepatitis D is given. HBsAg, hepatitis D, so HBsAg and hepatitis D can then infect the cells on its own. Wait. You need Hep B virus to be infected with hepatitis D. Hep D is non-functional when Hep D is given. GSAG Hep D can then infect the cells on its own. It is non-functional. Hep D is non-functional when Hep D is given. HBsAg, hepatitis B, and then hepatitis D can infect the cells on its own. Okay, so here's what that means. Okay, so you need hepatitis B virus to be infected with hepatitis D virus. Hepatitis D is non-functional when hepatitis D is given. HBsAg and hepatitis D, when given together, can infect the cells on its own. This is how hepatitis D becomes functional. So hepatitis D works through complement. Hepatitis D infects the body through complementation. Okay. Hepatitis D infects body through complementation. D infects body through complementation. Next we have phenotype mixing. So phenotype mixing occurs when simultaneous infection occurs with simultaneous infection of a cell with two viruses. For progeny 1, which is this right here, progeny 1, genome of virus A can be partially or completely coded, forming a pseudovariant with the surface proteins of virus B. Type B protein coat determines the tropism, which is the infectivity of the hybrid virus. Progeny from subsequent infection of a cell by progeny 1 will have a type A coat that is encoded by its type A genetic material. Okay. Alright, so what this is saying is that proteins, the surface proteins are, remember we said it's those little projections on the outside of the enveloped virus. So proteins are the outside coating of one virus. They coat the genotype of the other virus. So if you see right here, what they're saying is for progeny 1, which is this, the genome of virus A, the genome of virus A can be partially or completely coded. Yeah, the genome is the stuff on the inside, is the DNA. So it's, see that in the progeny, the blue stuff is in progeny 1. It can be completely coded with the proteins of virus B. So the blue is coded by the green. So virus A, the DNA is on the inside and the coding of B is on the outside. Okay, type B protein code determines the infectivity of the hybrid. Type B protein coat. So the coat determines the infectivity of the baby, which is the progeny. Okay, and the progeny from subsequent infection of a cell by progeny 1 will have a type A coat that is encoded by its type A genetic material. So when this makes another baby, it will have the virus A coat, like the coat of type A. So right here, but that's why this is blue. See this? Phenotype mixing. So at first, virus A genome is transferred here, and then it's coded by the virus B. Then when this progeny 1 makes another child, the DNA is transferred over and the coat is of the first one, the virus A. Or you can just think of it like, well, type A, that is, yeah, the genetic material transfers over, and then the coat will be of whatever DNA this is. So this blue, the blue coat, the blue DNA, it's a DNA is blue, and the coat will be of that of the DNA that's on the inside, which is why this is also blue. I hope that made sense. That's phenotype mixing. Okay, next. So DNA viral genomes. There will be okay. All DNA viruses have double-stranded DNA genomes except for parvovirus, which is single-stranded DNA. Okay. All are linear except papilloma, polyoma, and hepatitis D. Hepat. Hepadnavirus. So hepatnavirus are like hepatitis B virus. Okay. All right. Those are circular. All our DNA, all our double-stranded DNA, like ourselves, except part of a virus, which is parvovirus, which is single-stranded DNA. Okay. RNA viral genomes. All RNA viruses have single-stranded DNA. All RNA viruses have single-stranded RNA genomes except Viridae, which is double-stranded RNA. Positive-stranded RNA viruses. I went to retrovirus, togovirus, flavivirus, coronavirus, herpesvirus, calcivirus, and picornavirus. Okay. So all RNA viruses have single-stranded RNA genome except for reovirus, which is double-stranded RNA. The positive-stranded RNA viruses are retrovirus, togovirus, flavivirus, coronavirus, herpesvirus, calcivirus, and picornavirus. Okay, so all RNA viruses are single-stranded RNA except for this one, reovirus, which is double-stranded RNA, and the positive-stranded RNA viruses are retrovirus, togovirus, coronavirus. Okay. Naked viral genome infectivity is purified nucleic acids of most double-stranded DNA viruses except for poxvirus and hepatitis B virus, and positive strand of single-strand RNA viruses are infectious. Naked nucleic acids of negative strand, single-stranded RNA, and double-stranded RNA viruses are not infectious. They require polymerase containing the complete virus. Okay. Purified nucleic acids of most double-stranded DNA viruses except poxvirus and hepatitis B. Okay, purified nucleic acids of most double-stranded DNA viruses except poxvirus and hepatitis B and positive strand single-strand RNA viruses are infectious. Okay, are infectious. Okay. naked nucleic acid, negative strand, single strand RNA, and double strand RNA. Viruses are not infectious. So nucleic acids of most double-strand DNA virus and positive strand single-stranded RNA virus are infectious. The naked nucleic acids of negative-stranded single-stranded RNA and double-stranded RNA viruses are not infectious. Now, viral envelopes. Generally, enveloped viruses acquire the envelope from plasma membrane when they exit the cell. Generally, enveloped viruses acquire their envelope from plasma membrane when they exit the cell. Enveloped viruses acquire their envelope from plasma membrane when they exit from cell. Generally, enveloped viruses acquire their envelopes. Generally, enveloped viruses acquire their envelope from plasma membrane. Generally, enveloped viruses acquire their envelopes from plasma membrane when they exit the cell. Okay. Exceptions include herpes virus, which acquire envelope from the nuclear membrane. Okay, remember that. Okay. So that's important. Generally, envelope viruses acquire their envelopes from plasma membrane when they exit the cell. Exceptions include herpes virus. Herpes virus acquire envelope from nuclear membrane. Okay. Now, naked ones, which are the ones that are not infectious, include papillomavirus, adenovirus, parvovirus, polyomavirus, calcivirus, piconavirus, revirus, and herpesvirus. Okay. Envelope DNA virus have helpful. Okay. Envelope viruses get their envelope virus get their envelope from the plasma membrane when they leave the cell. Except for the herpes virus, which get their envelope from the nuclear membrane. Naked, non-enveloped virus include papillomavirus, adenovirus, parvovirus, polyomavirus, piconavirus, reovirus, herpes virus, herpesvirus. What is herpesvirus? Oh, herpesvirus, hepatitis E. Okay. Next, DNA virus characteristics. Some general rules. DNA virus characteristics are DNA, herpes, adenovirus, pox, parvovirus, papilloma, polyomavirus. They're double-stranded except for parvovirus, which is single-stranded. Single-stranded DNA. Okay. DNA viruses have linear genomes, except for the papilloma and polyoma. The papillomavirus, which causes HPV, and polyomavirus. What is polyoma? Yeah. Okay. So polyoma, it also causes some strain of HPV. Okay. It causes JC virus and BK virus. So JC virus is the thing that causes progressive multifocal leukoencephalopathy, and that's usually seen in AIDS patients. That is caused by polyoma virus. Okay. All right. Now, have linear genomes except papilloma and polyoma and HepaDNA. Okay. Have linear genomes. So all DNA viruses have linear DNA except papilloma and polyoma, which are circular, and HepaDNA, which is circular also. DNA viruses are isosahedral, except for the pox virus. DNA viruses replicate in the nucleus, except the pox, except the pox virus, which carries its own DNA-dependent RNA polymerase. Okay. DNA viruses all replicate in the nucleus except for the pox virus. Pox is out of the box. Okay. So herpes virus, it is enveloped. It's double-stranded and linear. Medical important. See herpes virus entry. Pox virus. Yeah, it's enveloped, but it's not in the nucleus. Like, it doesn't replicate in the nucleus. Okay, so yeah, pox virus is enveloped, double-stranded linear. It's the largest DNA virus. Smallpox, it causes smallpox, which is eradicated worldwide by the use of live attenuated vaccine. Cowpox, milkmaid blisters. What is cowpox? Okay, so milkmaids would get sores on their hands from milking sick cows. Okay, cowpox. Molluscum contagiosum is flesh-colored papill with central umbilication. Okay. Okay, this is Molluscum contagiosum is caused by smallpox. See this? Okay. Hepat virus, it's enveloped. It's partially double-stranded and circular. Okay, this is what causes hepatitis B. It can be acute or chronic hepatitis. It is not a retrovirus but has reverse transcriptase. So remember this, reverse transcriptase. Okay. Adenovirus, it is not enveloped. It is double-stranded and linear. This causes febrile pharyngitis, which is figure A, where's figure A? Febrile pharyngitis, oh, it's over there. This is right here, sore throat. You see sore throat. Okay. Acute adenovirus causes acute hemorrhagic cystitis, pneumonia, conjunctivitis, pink eye, gastroenteritis, myocarditis. So basically, Adena has pink eye and tummy issues. It's caused by adenovirus. Yeah, pink eye and gastroenteritis. Just they'll have like everything. They'll have lung issues like coughing and fever, and they have stomach issue also. So it's caused by adenovirus. It's a DNA virus. Okay, they like to ask this, and they like to mix enterovirus, adenovirus, norovirus. So make sure you remember this. Adenovirus causes pneumonia, gastroenteritis, and pink eye, and cough, fever, and sore throat. Okay. Papillomavirus is a DNA virus. It is not enveloped. It is double-stranded and circular. Papillomavirus causes HPV, which also causes warts. HPV causes warts. The one that causes warts are serotype 1, 2, 6, and 11. SIN, cervical intraepithelial neoplasia, is also caused by HPV. And this also causes the cervical cancer, and the strains that cause cervical cancer is 16 and 18. You gotta remember that, okay? 16 and 18. There's a lot of clinical significance. 16 and 18. HPV, this warts won't kill you. It's contagious, but it won't kill you. This other one, the 16 and 18, they're evil to kill you. So just think like the higher the number, the worse the situation for HPV. At least in this case, 16 and 18. Polyomavirus, it is not enveloped. It is double-stranded and circular. This is what causes the JC virus, which is progressive multifocal leukoencephalopathy in HIV patients. This polyomavirus also causes BK virus, which is seen in transplant patients. Commonly targets the kidneys. Commonly targets the kidneys. JC, I don't know. These like mnemonics, they don't work for me. I just remember polyomavirus. Polyomavirus causes JC and BK virus. Okay, polyomavirus. So JC virus here. JC virus. It's a disease of oligodendrocytes. Destroys the white matter of the brain. Okay. This right here. See this? Okay. That's what JC virus looks like. Next. Okay, I'm just gonna make a note of this. Okay, next, paravirus. Okay, paravirus is it is not double-stranded. Okay, it is not double-stranded. I'm sorry. Whoa. Okay. Yeah, well, it is not double-stranded. It is single-stranded. Okay, it is not enveloped. Paravirus is not enveloped, and it is not double-stranded. It is single-stranded and linear. Remember that, single-stranded and linear. Okay. They like to ask, like, they like to describe somebody that has a B19 virus and the slap cheek rash in kids. Or they like to say, like, a teacher was standing a long time and yeah, standing a long time describes arthritis. And then they ask me, I think she's pregnant. There's some question like that, and then they describe this rheumatoid arthritis, and then you know it's paravirus, and they give you is it double-stranded, single-stranded, what, you know, like that. So you have to know single-stranded and linear, single-stranded and linear. Smallest DNA virus. What is the largest DNA virus? It's the pox virus. Okay. All right. So paravirus causes B19 virus, which is aplastic crisis in sickle cell disease. You get a slap cheek rash in children, erythema infectiosum, This is what erythema infectiosum is. The kids look like somebody slapped them to this fifth disease. Okay. So yeah, B19 virus that causes aplastic crisis in sickle cell disease, slap cheek rash in children, which is known as erythema infectiosum or fifth disease. Paravirus infects RBC precursors and endothelial cells, and which leads to RBC destruction. This leads to hydrops vitalis and death in the fetus. Okay, hydrops vitalis. If the mom has paravirus, this can happen in the kid. Hydrops vitalis. This is hydrops vitalis. So it's childhood dead, but like once you have hydrops vitalis, wow, well that's extremely rare. Oh, that's amazing. Praise God. Okay, but in general, a lot of the situations, the baby is dead. Okay. Okay. Yeah, this is hard to look at right here. So that's hydrops vitalis. Yeah. Has fluid accumulation. That's the baby is swollen. That's what paravirus causes. Paravirus is serious. Okay. So yeah, causes hydrops vitalis and death in the fetus. PRRBC aplasia and rheumatoid arthritis like symptoms in the adults. RBC aplasia. Yeah, it's when the bone marrow doesn't make enough. Doesn't make enough RBC. So the mom has rheumatoid like arthritis. So yeah, basically, if you have a pregnant lady, she has pancytopenia, and she has rheumatoid arthritis like symptoms. Um, think paravirus. Okay. Okay. Herpes virus, enveloped double-stranded linear virus. Yeah, herpes virus, enveloped double-stranded and linear viruses. Herpes virus, enveloped double-stranded and linear viruses. Okay, what you have to know is herpes simplex 1. The route of transmission is through respiratory secretions and saliva. It causes herpes simplex 1, HSV1, causes gingiovostomatitis, keratoconjunctivitis, herpes labialis, which is cold sores, and then herpetic whitlow on the finger, temporal lobe encephalitis, and esophagitis, erythema multiform. The node is most commonly latent in trigeminal ganglia. HSV1 most common cause of sporadic encephalitis can present as altered mental status, seizures, and/or aphasia. Okay. All right. So basically, what you have to know about this is that HSV1 is fed by saliva. If somebody sneezes, respiratory secretions. I mean, you had to. I mean, so many people have this because it's so easily spread. But yeah. So gingiostomatitis, keratoconjunctivitis. This is figure A right here. Right here, that's figure A. This is caused by HSV1. This is ulcers in the cornea on fluorescein slit lamp exam. That's ulcers, okay, in the eye, okay? Alright. Now, herpes labialis is B. Right here, this is cold sores. It's caused by HSV1. And then you have a herpetic whitlow on the finger. Which is common in dentists because they keep touching people's mouths. Okay, herpes whitlow. Let me show you. Herpetic whitlow. This is herpetic whitlow. Okay, right. And dentists, a lot of dentists can get this because they're touching people's mouths. Yeah, so remember that. Okay. HSV1 can also cause temporal lobe encephalitis. You have to remember that temporal lobe encephalitis is very important. Temporal lobe encephalitis is temporal lobe. Okay, this is. Say this. Temporal lobe encephalitis. HSV encephalitis is an invasive infection of the central nervous system. Up to 5% of patients. Seen up to 5% of patients. So this Babies can get this if somebody with a cold sore comes and kisses your child, your newborn, because they don't have a they don't have an immune system to fight. So basically, if somebody with a cold sore comes and kisses your newborn when they come to see the baby, and baby can get temporal lobe encephalitis. Don't let anyone kiss your kid. Because it's one, it's so common. Like herpes, like cold sores are so common. People don't know, right? So they come and kiss, like because of infection, but it can be extremely fatal for the baby. Yeah. In fact, I read a case about this. They didn't understand what happened, like why the baby was sick. Not sure. That's something else. Alright, so temporal encephalitis is caused by HSV1, but the kids, this kid died of meningitis caused by HSV1. But yeah, just remember Remember both meningitis and temporal lobe encephalitis. Okay. First aid is given temporal lobe encephalitis, but remember both. That's why I don't kiss your kid. Okay. So that's there. Then HSV1 is in the trigeminal ganglia. That's where it sleeps. Most common cause sporadic encephalitis can present as altermental status, seizure, and europhagia. Herpes simplex 2. Oh, erythema multiforme. Multiforme. This is erythema multiforme caused by HSV1. Okay. This is erythema multiforme in a baby. Okay. It presents erythema multiforme presents 1 to 2 weeks after the infection of HSV1. Okay. Now next, herpes virus. The other type of herpes virus is HSV2. You get this by sexual contact or through perinatal transmission, which is mom to baby transmission. Okay. And then clinical significance herpes genitalis, which is C. genitalis. Let's see. What is this right here? This is herpes genitalis. Alright, maybe that's not a good picture. Herpes genitalis. As yeah, so that's the scene. It's genital source. Genital source caused by HSV2. HSV2. Now it can also cause a neonatal herpes. Okay. This is there's painful inguinal lymphadenopathy. Herpes simplex 2 is most commonly latent in sacral ganglia. So HSV1 is in the trigeminal ganglia, that's where it sleeps. HSV2 is in the sacral ganglia, okay? So HSV2 lies dormant in sacral ganglia. Just think HSV2 causes genital herpes and your sacrum is closer to your genitals. Where's your sacrum? Yeah, so Just think your sacrum, this yellow thing, is closer to your genitals. That's how you learn medicine. You gotta make connections. Weird, funny ones, too. Even the ones that don't make any sense. If it helps you, it helps. That question can make or break you, okay? Alright, so now, okay, I see. HSV2, it causes viral meningitis, more common with HSV2 than HSV1. HSV1 causes temporal lobe encephalitis. HSV1 causes temporal lobe encephalitis. HSV2 causes viral meningitis. Yeah. So we mentioned it's painful inguinal. There is painful inguinal lymphadenopathy. HSV2. So we have to know HSV2 causes aseptic meningitis. Yeah, it causes aseptic meningitis in adolescents and adults. You see TZANK zinc smear showing multinucleated giant cells, but it's PCR test now. The treatment is acyclovir or valcyclovir for treatment. Okay. All right, now we're gonna go into the other viruses, other DNA viruses. So for herpes virus, you have to know the intranuclear inclusion bodies are Cowdrey bodies. Just remember that in the intranuclear inclusion bodies are Cowdrey bodies, which are right here. Cowdrey bodies. Cowdrey bodies. These are Cowdrey bodies. Okay. These browns right here. Alright, next. Varicella zoster. Virus is this. Varicella zoster virus is a route of transmission is through respiratory secretions, contact with fluid from vesicles. Okay, so if a vesicle pops and you come into contact with that, you can get that. The clinical significance is varicella zoster virus, which causes chickenpox and shingles. Varicella zoster virus also causes encephalitis and pneumonia. Most common complication of shingles is post-herpetic neuralgia. Varicella zoster virus is latent in dorsal root or trigeminal ganglia. CN, cranial nerve, 5.1, which is okay. So there's mandibular, there's maxillary, there's ophthalmic, right? So let me just look, CNV1. Yeah, that's ophthalmic division of the trigeminal nerve. It's latent in the dorsal root or trigeminal ganglia. CN5.1, so the ophthalmic division of the trigeminal nerve. Involvement can cause herpes zoster ophthalmicus. Okay. So post-herpetic neuralgia is a long-lasting pain in the areas of your skin where you had shingles. Okay. So shingles is already super painful. So if you have shingles, a complication of that is post-herpetic neuralgia, which is pain where the rash was. Okay. Alright, next. Notes is latent in the dorsal root or trigeminal ganglia. Ophthalmic division of trigeminal branch involvement can cause herpes zoster ophthalmicus. Okay, herpes zoster ophthalmicus is a viral involvement of the ophthalmic division of the trigeminal nerve. So this causes pain and tingling of the forehead, blisters on the forehead and nose, eye ache, redness, light sensitivity, eyelid swelling. Alright, so yeah, it causes like a rash wherever the rash on the forehead. Wherever the trigeminal nerve supplies, it causes issues there. So, rash and tingling on forehead. Rash and tingling on forehead. Okay, latent dorsal root trigeminal ganglia, C and 5-1 branch. Involvement can cause herpes zoster ophthalmicus. Okay. So, figure D is chickenpox. Okay, most commonly seen in children. Chickenpox, chickenpox, and then this other one, E is shingles. Shingles has a very distinct rash. It's super painful. That's what shingles looks like. It's seen in older people. Shingles. So basically, if you had chickenpox as a kid, you're at risk to get shingles later in life. The varicella zoster virus goes and hides in your dorsal root, or in the trigeminal ganglia. Then it reactivates when you're old and comes back as shingles and causes this rash seen in figure E. Alright? Okay. And a complication of the rash is post-herpetic neuralgia. Okay, next. Epstein-Barr virus, very important, always tested. Right, Epstein-Barr virus, it's caused of respiratory secretions through kissing mainly. It can be. It's known as the kissing disease. Mononucleosis, right? Mononucleosis is known as the kissing disease. Disease is common in kids, well, teens, and young adults, mainly people in college. They present with mono. It's passed by saliva. Mononucleosis causes fever, hepatosplenomegaly, which is seen in figure F. See this figure F right here? Hepatosplenomegaly, liver and spleen are both enlarged. So okay. So in mono you have fever, hepatosplenomegaly, pharyngitis, and lymphadenopathy, especially in the posterior cervical nodes, and you have to avoid contact sports until resolution due to risk of splenic rupture. Epstein-Barr virus associated with lymphomas. It causes 3 types of cancer. Epstein-Barr virus causes Burkitt lymphoma, and number 2, the cancer causes nasopharyngeal carcinoma. And number 3, Hodgkin's lymphoma. Okay. So Epstein-Barr virus causes non-cancerous lesion like oral hairy leukoplegia in HIV patients, and also causes cancer, which is the 3 types we just mentioned: Burkitt lymphoma, nasopharyngeal carcinoma, and Hodgkin's lymphoma. Nasopharyngeal carcinoma is usually in Asian adults, especially it's seen in Asian adults. Nasopharyngeal carcinoma. So this right here, this is the cancer, nasopharyngeal cancer. So basically it's cancer in your nasopharynx. Nasopharynx right here, that picture I just showed you, nasopharynx, oropharynx, laryngopharynx. So cancer right here, that is caused by EBV, Epstein-Barr virus, and Burkitt lymphoma. So let's see. I wonder why it's causing Asian people, Asian men. Okay, so that's that, and then Burkitt's lymphoma. Burkitt lymphoma. This is Burkitt lymphoma. You're never gonna forget this. Yeah. So Epstein-Barr causes Burkitt's lymphoma, which is jaw swelling. And then also causes Hodgkin's lymphoma, in which you see Reed-Sternberg cells. You have to know what Reed-Sternberg cells look like. Reed-Sternberg cells. This is Reed-Sternberg. It looks like allies right here. Reed-Sternberg legs. Reed-Sternberg cells, which is seen in Hodgkin's lymphoma. EBV causes endemic Burkitt lymphoma, where you see large jaw and swelling. Yes, and then it also causes oral hairy leukoplegia. You also see lymphoproliferative disease. You also see lymphoproliferative disease in transplant patients. You have to know Epstein-Barr virus infects the B cells through CD21. Okay, that's important to know. So what that means is EBV envelope glycoprotein binds CD21 to infect B cells. So CD21 is found on top of B cells, and then Epstein-Barr virus goes and attaches to CD21 to infect the B cells. Okay? Alright. Atypical lymphocytes on peripheral blood smear. Atypical lymphocytes on peripheral blood smear, which is G. So G, figure G right here. You see atypical lymphocytes on peripheral blood smear. Not infected B cells. These are not infected B cells, but reactive. Belle, stop. Stop crying. You know you don't want anything. Stop it. It's my cat, guys. Sorry. Alright. Now infected B cells. Bella. Come here. Come here, Bella. Come here, Bella. Come here, Bella. Oh. She just wants attention. Does your cat do this? When you go to her, she stops screaming. Yeah. Okay. So that's that. Yeah, it's a figure G. This is reactive cytotoxic CD8 positive T cells seen on blood smear. It is not B cells, okay? It's a reactive cytotoxic T cells seen on blood smear. In Epstein-Barr virus, you have a positive monospot test. You have a positive monospot test, which causes heterophile antibodies. Okay, positive monospot test. Heterophile antibodies detected by agglutination of sheep or horse RBCs. Okay, so basically Epstein-Barr virus activates the B cells to secrete heterophile anti-sheep RBC antibodies, and these RBC antibodies will clump sheep, horse, or RBCs. Okay, so yeah. Atypical lymphocytes on peripheral blood smear, non-infected B cells or reactive cytotoxic T cells. Positive monospot test. So what you see here is hetero antibodies detected by agglutination of sheep or horse RBC. So EBV activates B cells to secrete heterophile anti-sheep RBC antibodies, and therefore RBC antibody will clump sheep, horse, or RBCs. And therefore RBC, yeah. And therefore yeah. Heterophile antibodies detected. Heterophile antibodies detected by agglutination. Of sheep or RBC. So basically, the sheep or horse RBCs will clump together. Very serious. Okay, guys, I shall be back. Okay, we had a longer break because my cat decided she wants attention. So, yeah. Anyway. So in Epstein-Barr virus, you have a positive monospot test, which is when heterobial antibodies are detected by sheep or horse RBCs being clumped together. So EBV activates beta cells, B cells, to secrete heterophile anti-sheep RBC antibodies, and these RBC antibodies will clump sheep, horse, or RBCs together, and that's how you know you're working with the Epstein-Barr virus, right? Because this was always going to be monospot positive test. Okay. CMV is also extremely heavily tested. The biggest complication of mononucleosis is patient has a large spleen due to T cell proliferation, and you're always worried the spleen will rupture. So that's why they tell, especially college kids, don't be active in sports if they have mono. Don't be active in sports because you're worried about their spleen rupturing. Next, CME. This one is a congenital transfusion. Yeah, this is a congenital transfusion. You can get from sexual contact, saliva, urine, or organ transplant. This one it causes mononucleosis, but it is negative monospot test in immunocompetent patients. Infection in immunocompromised, especially pneumonia in transplant patients, and then you have esophagitis, AIDS retinitis, hemorrhage, cotton wool exudates, vision loss, and congenital CMV. So these are the diseases CMV causes. CMV in healthy patients will cause mononucleosis, and it will be a negative monospot test. It can also cause infection in immunocompromised patients, especially pneumonia in transplant patients, esophagitis, AIDS retinitis, hemorrhage, AIDS retinitis. In AIDS retinitis, you have hemorrhage, cotton wool exudates, and vision loss. What are cotton wool exudates? Cotton. So this is what that is. It looks like cotton. Let's see, CMV, cotton wool spots, hemorrhage. So this, what we said here. Oops. Hemorrhage, cotton wool exudates, vision loss, all seen in AIDS retinitis, hemorrhage, this red spot, hemorrhage, cotton wool, these white spots look like cotton. Alright? Next. Vision loss, it can cause congenital CMV. Infected cells have characteristic alloy intranuclear inclusions, like seen in figure H, right here. Intranuclear alloy inclusions. So that is CMV, okay? Don't forget that, that's important. Intranuclear inclusions. So these rounds, they're inside the nucleus. Okay, alloy, intranuclear, intranuclear inclusions seen in CMV. Okay, um, it's in mononuclear cells. It hides, sleeps in mononuclear cells. What are mononuclear cells? Mononuclear. Mononuclear cells are a mixture of various different types of cells and contain mostly different stem cells. Okay. So these are monoblasts, monocytes. Okay. Mononuclear cells. Monoblasts. So these are monoblasts, monocytes, monocytes. Okay, so these CMV hides in monoblast monocytes. Okay. The cotton wool exudates, vision loss. Okay. Human herpes virus 6 and 8 pass by saliva. Um, it causes roseola infantum. Um, roseola infantum. High fevers for several days that can cause seizures, followed by diffuse macular rash. Starts on the trunk and spreads to extremities. Right here. Starts on the trunk and spreads to extremities. 2-minute coffee break, okay? Okay. Yeah, Roseola infantum, also known as exanthum subitum, uh, causes high fevers for several days that can cause seizures followed by diffuse macular rash, which is a flattened rash, no bumps. Okay, it's a flattened red rash. It starts on the torso, then spreads to the arms and legs. Okay, so this is figure I right here. And figure I, that is the Roseola infantum. I'm going to write this down. Roseola infantum. I wrote it in pink because Roseola infantum, because it's seen in babies. Roseola infantum. Um, yeah, caused by herpes virus, herpes virus 6 and 7. Okay, so note this causes the roseola fever first, then a rosy rash later. So fever first, rosy rash later. HHV7, less common cause of roseola. Human herpes 8, spread by sexual contact. This is what causes Kaposi sarcoma, which is a cancer of the endothelial cells, which is seen almost always in HIV and AIDS patients, and also in transplant patients. Dark violaceous plaques on nodules representing avascular proliferation. J. This is a Kaposi sarcoma, okay, caused by human herpes virus 8. Kaposi sarcoma. Kaposi sarcoma, cancer of the endothelial cells. Okay. Kaposi sarcoma. Kaposi sarcoma caused by HHV8. HHV8. Okay. Also caused GI. Can also affect the GI tract and the lungs. This Kaposi sarcoma. Okay. Oh my gosh. Oh great. Good friend. Good too. Okay, now we have HSV identification. Um, so this is a viral culture for skin. Viral culture for skin and genitalia. CSF, cerebrospinal fluid, PCR for herpes encephalitis. Uh, SNAC test is made of an open skin vesicle to detect for multinucleated skin cells, which is seen in A. These arrows are pointing to multinucleated giant cells. Okay, so this is multinucleated giant cells. Um, multinucleated giant cells. Multinucleated giant cells. Multinucleated giant cells. Okay, multinuclear giant cells are seen on T-zinc test. Okay. Alright, commonly seen in HSV1 and HSV2 and rare zoster infection. And PCR skin lesions, they test of choice now. But no, they like to ask about the multinuclear giant cells seen on T-zinc speed test. Zinc test, okay. Alright. Okay. And then intranuclear eosinophil cautery A inclusions also seen in HSV1, HSV2, and rare zoster virus, like you said before. Okay. So viral culture, how to identify the HSV. Yeah, viral culture for skin and genitalia, CSF PCR for human herpes encephalitis. Okay. Yeah, so you do PCR of the cerebrospinal fluid. T-zinc test. That's a skin test. You take the fluid out of the open vesicle and you look to see if they have multinucleated giant cells. And this is an HSV1, HSV2, and as well as zoster virus. But now they do PCR. Also, intranuclear eosinophilic cautery A inclusions, also seen in HSV1, 2, and varicose zoster virus. So cautery A inclusions are the inclusion bodies seen in HSV 1, 2, and varicose zoster virus. Receptors used by viruses. Okay, this is extremely high yield. You have to know they love to ask us. They love. So CMV. CMV uses integrins to bind to cells. Okay, CMV uses what? Integrins to bind to the cells to infect them. EBV binds to CD21. Remember we said CD21 is on top of B cells, so CD21. Yeah, EBV binds to CD21. EBV, oops, CD21. Okay. See, so this is the Epstein-Barr virus. This is the B cell. It's chilling normally in our body, and naturally the CD21 is on top of the B cells. EBV comes directly for the CD21 and affects the B cells. How evil. Okay. Anyway, so that's that. CMV integrins. Let me show you this too, because I've never actually seen it. CMV integrins. Integrins binding. Okay, so where is it? CMV. Well, I can't find a picture of it. Okay, so so heparin sulfate right here. CMV comes and binds, I guess. Okay, imagine this is CMV. CMV comes and binds the cell through heparin sulfate. That's how we remember this. Okay. All right. Because can't find a proper picture. HIV binds to CD4, CXCR4, CCR5. Okay, remember that. HSV binds cells using CD4, CXCR4, CCR5. Pyrovirus B19 binds through P antigen, which is normally on the RBC. So the P antigen is normally on the RBCs. Pyrovirus comes and binds to the P antigen, therefore infecting RBCs, causing pancytopenia and total destruction of RBCs. Okay? Remember rabies. Rabies uses the nicotinic acetylcholine receptor to bind the cells in our body. Rabies is 100% fatal once the symptoms show. Okay, there's nothing you can do. So rabies is a very sad disease. I saw a few videos of kids with rabies, and honestly, I couldn't watch it. It was very, very sad. Rabies, yeah. So it uses a nicotinic acetylcholine receptor to bind to the cells in our body. Rhinovirus uses ICAM1, ICAM1. I guess you can remember that. I came to see the rhino. I guess that's a good one. Rhinovirus. Think of like the safari. I think of rhinos. I think of safari, and I came to see the rhino at the safari. People go. That's what people do. It's a tourist destination, right? Came to see rhinos. All right. Now we're done with DNA viruses. Next, we're gonna go to RNA virus in the next video. See you then.